Imagine standing at the edge of a vast canyon. The rock face before you showcases a perfect timeline, billions of years of Earth's history etched into stone. Layer upon layer, each representing thousands or millions of years of geological change. Now imagine that somewhere in those layers, barely visible to the naked eye, lies a thin stripe that represents the entire span of human existence. Yet, in that microscopic sliver of time, something remarkable happened. A species emerged that would eventually build skyscrapers, split atoms, and send radio signals to the stars. We like to believe our ascent was obvious, the inevitable march from knuckle-walking ape to upright thinker. But what if our most important evolutionary adaptations weren't the obvious ones? What if the true masterpieces of human design are invisible even to us who carry them? Dr. Elena Takahashi stands in her laboratory at Stanford University, holding what appears to be an ordinary vial of blood. The revolution, she says, is happening at the molecular level. We've been looking at bones and brains for centuries, but the real story of human evolution is written in biochemistry most people never think about. This is the story of humanity's hidden upgrades, the subtle, often invisible adaptations that made us who we are. Forget opposable thumbs and large brains for a moment. Those are the obvious features, the ones we celebrate in museums and textbooks. But beneath our skin, inside our cells, and woven into our biochemistry are modifications so elegant and powerful that they remained largely undiscovered until the 21st century. These are the secret architects of human potential, and they might change how you understand what it means to be human. The story begins not with humans, but with a crisis that occurred billions of years ago. A catastrophe so profound that it nearly wiped out all life on Earth. Approximately 2.4 billion years ago, certain bacteria developed the ability to perform photosynthesis, releasing oxygen as a waste product. This oxygen was toxic to most life forms that had evolved in the previously oxygen-poor environment. Scientists call this the Great Oxygen Catastrophe, the first mass extinction event in Earth's history. Oxygen was essentially pollution, explains paleobiologist Dr. Victor Williams. Imagine if the air around you suddenly became filled with something that corroded your body from the inside out. That's what happened to early life forms when oxygen levels rose. But from this catastrophe came opportunity. Organisms that could tolerate oxygen had a new energy generation method at their disposal. Aerobic respiration produces about 16 times more energy than anaerobic processes. Life forms that adapted to use oxygen gained a massive energy advantage. Fast forward to humans. We tend to think our relationship with oxygen is simple. We breathe it in, our cells use it, and we breathe out carbon dioxide. But the human oxygen processing system contains sophistication that remained hidden from science until recently. In 2009, researchers at the University of Cambridge made a startling discovery. Human hemoglobin, the protein in red blood cells that carries oxygen, has undergone subtle but critical modifications compared to our primate relatives. These modifications allow our blood to deliver oxygen more efficiently during sustained physical activity. It's not just about how much oxygen you can take in, Dr. Takahashi explains. It's about precision delivery, getting the right amount to the right tissues at the right time. This precision is particularly evident in human endurance activities. Studies of persistence hunters in the Kalahari have shown that humans can maintain moderate running speeds for hours, long after most other mammals, including horses and antelope, have collapsed from heat exhaustion. What makes this possible isn't just our bipedal stance or lack of fur. It's the remarkable efficiency of our oxygen delivery system. Human hemoglobin has developed subtle structural changes that allow it to maintain optimal oxygen binding properties across a range of pH levels, temperatures, and metabolic states. Think of it as having a smart delivery system versus a basic one, says physiologist Dr. Ethan Thompson, demonstrating with a model of hemoglobin. Other primates' oxygen delivery is like a simple pipe. It works, but it's not adaptable. Human hemoglobin is more like a smart irrigation system that can adjust flow based on changing conditions. This invisible upgrade helped fuel the metabolic demands of our growing brains, which consume 20% of our oxygen despite being only 2% of our body weight. But it also provided another unexpected advantage: the ability to sustain physical activity for extraordinary periods. The archaeological record shows that early humans didn't rely on superior speed or strength to hunt. Instead, they practiced persistence hunting, tracking animals until they collapsed from heat exhaustion. This method depends precisely on the kind of optimized oxygen delivery system that humans evolved. When we look at the Tarahumara people of Mexico who can run 200 miles without stopping, or Kenyan marathoners who dominate long-distance running, we're seeing the expression of this hidden adaptation. Dr. Williams notes, they're not genetic outliers. They're humans utilizing the full potential of an evolutionary adaptation we all possess. But this raises a puzzling question. If our oxygen processing system is so sophisticated, why aren't we aware of it? Why doesn't it feature prominently in our understanding of what makes humans special? To understand why some of our most important adaptations remain hidden from our awareness, we need to explore what anthropologists call the invisible present, the tendency for humans to notice sudden changes while remaining oblivious to gradual ones. We're excellent at detecting immediate threats or changes, explains cognitive anthropologist Dr. Victoria West. A sudden movement in the grass might be a predator. A sudden change in someone's facial expression might indicate anger. But gradual changes, those happening over generations or lifetimes, often escape our notice entirely. This perceptual blind spot creates an interesting paradox. Some of our most important evolutionary adaptations are invisible to us precisely because they work so well and have been with us for so long. They form the background conditions of our existence rather than features that demand our attention. Consider temperature regulation, an area where humans have developed remarkable specialization. Most mammals maintain a constant internal temperature, but human thermoregulation contains subtle sophistications that went unnoticed until recently. Humans have approximately 2 to 4 million sweat glands distributed across our bodies. Dr. Thompson explains, that's far more than necessary for basic cooling. The density and distribution pattern of these glands represent a major evolutionary investment. This investment paid extraordinary dividends. Unlike most mammals that pant to cool down, a method that works but interferes with breathing during exertion, humans cool through sweating, allowing simultaneous cooling and oxygen intake during physical activity. Moreover, human sweat has a unique composition. Studies comparing human sweat with that of other primates reveal that ours is remarkably dilute, containing lower concentrations of salts and proteins. This seemingly minor detail proves crucial for extended cooling. If you sweat for hours with high salt concentration, you quickly develop electrolyte imbalances. Dr. Takahashi explains, our dilute sweat allows for extended cooling without dangerous mineral losses. The sophistication extends to the neural control of sweating. Human sweat response begins before our core temperature rises significantly, a predictive rather than reactive system. This anticipatory sweating gives us a crucial head start on temperature regulation. It's like having a smart thermostat versus a basic one, Dr. Williams says. Most animals react to overheating. Humans anticipate it. Our bodies begin cooling before we reach problematic temperatures. These thermoregulatory adaptations, working in concert with our oxygen delivery system, helped create what anthropologists call the human thermal niche, our unique ability to remain active in hot conditions that would incapacitate most mammals. Yet, despite its sophistication, most humans throughout history remained unaware of these systems. We notice when we sweat, but the intricate mechanisms controlling that process, the millions of precisely tuned glands, the predictive neural circuits, the carefully balanced composition, remained invisible until scientific investigation revealed them. This pattern of invisible sophistication appears repeatedly throughout human biology. Our most critical systems often operate below the threshold of our awareness, shaping our capabilities without announcing their presence. Perhaps nowhere is evolutionary sophistication more evident yet invisible than in human digestive biochemistry. While we share the basic digestive architecture with other mammals, subtle changes in our digestive enzymes and detoxification systems have dramatically expanded the range of foods humans can consume. Dr. Emily Turner, a biochemist at University College London, has spent decades studying these digestive adaptations. We've identified at least 46 enzymes unique to humans or significantly modified in humans compared to our closest relatives, she explains. These molecular innovations greatly expanded our dietary options. One striking example involves alcohol metabolism. Humans possess a variant of the ADH4 enzyme that processes ethanol approximately 40 times more efficiently than the version found in other primates. This adaptation appeared approximately 10 million years ago, long before humans began deliberately producing alcoholic beverages. The timing suggests this enzyme variant evolved to help our ancestors consume fallen fruit that had naturally fermented, Dr. Turner notes. This would have provided access to high-calorie food sources that other animals avoided because of the alcohol content. But alcohol metabolism is just one example of our expanded detoxification capabilities. The human liver produces an array of enzymes specifically adapted to neutralize plant toxins. These enzymes, collectively known as cytochrome P450 enzymes, show significant differences between humans and other primates. Plants are engaged in chemical warfare, Dr. Takahashi explains. They produce thousands of compounds specifically designed to deter animals from eating them. Our enhanced detoxification systems allow us to consume plants that would poison other animals. This biochemical sophistication helped humans become one of the most adaptable omnivores on the planet. From the Arctic to the Kalahari, humans can subsist on dramatically different diets because our digestive chemistry can handle an extraordinary range of foods. The story becomes even more fascinating when we examine human starch digestion. Humans produce significantly more salivary amylase, an enzyme that breaks down starches, than other primates. Moreover, the number of amylase gene copies varies among human populations in direct correlation with historical starch consumption. Populations with traditionally high starch diets often have more copies of the amylase gene, explains genetic anthropologist Dr. Nathan Rivera. We're seeing molecular evidence of dietary adaptation occurring within the human lineage. These digestive adaptations played a crucial role in human brain evolution. The brain is metabolically expensive, requiring stable glucose supplies. Our enhanced ability to extract nutrients from diverse food sources helped fuel our growing neural complexity. There's a direct connection between our digestive biochemistry and our cognitive capabilities, Dr. Williams observes. The invisible chemistry happening in our digestive systems helped make our visible mental abilities possible. Yet despite their importance, these digestive adaptations operate entirely below our conscious awareness. We know when we're hungry or full, but the sophisticated enzymatic cascades that enable our dietary flexibility remain hidden from direct experience. This invisibility extends to another critical area of human biochemistry, one that might have played an even more significant role in our cognitive evolution. In 2019, a team of geneticists at MIT made a discovery that sent ripples through the scientific community. They identified a uniquely human gene called Notch2NL that appears to delay neural maturation, extending the period when brain cells proliferate and form connections. This gene, present only in humans and not in our closest relatives, essentially keeps brain development in an extended growth mode, allowing for the formation of more complex neural networks. It's like having construction workers stay on site longer, explains neuroscientist Dr. Sarah Chun. If you give the brain more time to develop, you can build more complex structures. This discovery highlighted a pattern that scientists had been gradually uncovering. Many of the most important adaptations in human evolution involve not just the adult form of structures, but the developmental processes that create them. Human brain development follows a unique trajectory. Our brains continue significant development after birth, a pattern called neoteny, or the retention of juvenile features into adulthood. This extended development allows our neural connections to be shaped by experience and learning rather than being hardwired from birth. Most animals are born with brains largely pre-programmed for their ecological niche, Dr. Chen explains. A gazelle can stand and run shortly after birth because its brain comes with those circuits already established. Human brains, by contrast, are remarkably unfinished at birth. This unfinished quality of the human brain might initially seem like a disadvantage. Human infants are helpless for years, requiring extensive parental care. But this developmental pattern allowed for something revolutionary: brains that could be shaped by culture and learning rather than genetic programming alone. The molecular mechanisms controlling this developmental trajectory remained mysterious until recently. Advanced genetic analysis has now identified an array of genes unique to humans that regulate neural development. These include genes that control the rate of neural progenitor cell division, the migration of neurons to their final positions, and the formation of synaptic connections. We've found at least 35 genes unique to humans that influence brain development, Dr. Takahashi explains. Some emerged through gene duplication, others through modifications to regulatory regions. Together, they create a developmental program unlike anything in other species. These molecular innovations helped create brains with extraordinary plasticity, the ability to reorganize in response to experience. Human neural plasticity extends far beyond that of other primates, allowing us to acquire new skills throughout life. If you examine the molecular machinery governing synaptic plasticity, the physical basis of learning, you find human-specific adaptations, notes Dr. Thompson. Our neurons can form and reorganize connections more readily than those of other primates. This enhanced plasticity explains an observation that had puzzled scientists. Humans can recover from brain injuries that would be permanently debilitating in other species. After stroke or traumatic injury, human brains can sometimes rewire to restore lost functions, a capacity for reorganization that exceeds that of our closest relatives. These neural adaptations operated synergistically with our extended childhood. Human children remain dependent on adults for an unusually long period, creating an extended learning window. During this period, our highly plastic brains absorb language, cultural knowledge, and social norms. We essentially co-evolved long childhoods and plastic brains, Dr. Williams explains. Each adaptation reinforced the value of the other, creating a powerful feedback loop that accelerated cultural evolution. This extended developmental window created another unique human characteristic, one that would prove crucial for our species' success. In 1997, anthropologist Robin Dunbar proposed that human social groups are constrained by our cognitive capacity to track relationships. He suggested that humans can maintain meaningful social relationships with approximately 150 individuals, a limit imposed by the computational demands of social cognition. This observation opened a fascinating question. What cognitive adaptations allow humans to maintain social groups far larger than those of other primates? The conventional answer focuses on our larger brains, explains cognitive scientist Dr. Veronica Wallace. But brain size alone doesn't explain our social capabilities. Something more subtle is happening in the neural circuitry dedicated to social cognition. Recent neuroimaging studies have revealed that humans possess enhanced development in brain regions dedicated to understanding others' mental states, what scientists call theory of mind. These regions, including the temporoparietal junction and parts of the prefrontal cortex, show unique patterns of connectivity in humans compared to other primates. It's not just that these regions are larger, Dr. Wallace explains. They show different patterns of activation and connectivity. Human social cognition isn't just scaled-up primate social cognition, it's qualitatively different. This difference becomes particularly evident in studies of human infants. By 14 months of age, human babies already demonstrate awareness that others have beliefs and intentions different from their own, a cognitive milestone that chimpanzees reach only partially, even as adults. The neural basis for this enhanced social cognition includes specialized cell types and circuits. Von Economo-neurons, a cell type particularly abundant in humans, are concentrated in regions involved in social cognition and self-awareness. These specialized cells, with their unique shape optimized for rapid signal transmission across distant brain regions, facilitate the integration of social information. These neurons essentially allow different parts of the brain to rapidly communicate about social situations. Dr. Chen explains. They're like dedicated information highways for social processing. But our social adaptations extend beyond neural architecture to include biochemical specializations as well. Humans show unique patterns in the production and receptor systems for oxytocin and vasopressin, hormones that regulate social bonding and trust. Human oxytocin receptors show subtle structural differences that alter binding properties, explains neuroendocrinologist Dr. Robert Patel. These molecular modifications change how strongly and under what circumstances social bonding occurs. These neural and biochemical adaptations created something unprecedented. A species capable of forming cooperative groups of extraordinary scale and complexity. While other primate species live in groups of at most a few hundred, human societies can organize millions of individuals in coordinated activity. This social coordination enabled perhaps the most transformative adaptation of all, one that occurred not within our bodies but between our minds. In the northern Spanish cave of El Castillo, a simple red dot painted on the wall has been dated to at least 40,800 years ago, making it one of the oldest known pieces of human artwork. What makes this simple marking revolutionary isn't its artistic sophistication but what it represents. The human capacity for symbolic communication. That dot means something, Dr. Williams explains, pointing to an image of the cave painting. We can't know exactly what it meant to its creator, but we know it was intended to convey meaning to other humans. That's the essence of symbolic communication. This capacity for symbolism, using one thing to represent another, enabled an evolutionary innovation of unprecedented power. Cumulative culture. Unlike other species whose behavioral innovations typically die with the individuals who discover them, humans can preserve and build upon knowledge across generations. Cultural transmission created a second inheritance system alongside our genes, explains cultural evolutionist Dr. Evelyn Torres. Information could now evolve and accumulate outside of biological evolution. This cultural transmission system dramatically accelerated the pace of human adaptation. Genetic changes typically require many generations to spread through a population. Cultural innovations, by contrast, can spread within a single generation. The neural foundations for this cultural capacity include our specialized language areas, regions like Broca's and Wernicke's areas that show unique development in humans. But even more important is the distributed network of regions that work together to support symbolic thinking. Language isn't localized to one brain area, Dr. Chen explains. It emerges from the coordinated activity of many regions, including those involved in motor control, auditory processing, and conceptual representation. This distributed architecture creates a system greater than the sum of its parts, one capable of generating and manipulating an infinite variety of meaningful symbols. The human capacity for recursion, embedding meanings within meanings, allows us to communicate ideas of extraordinary complexity. Recent genetic studies have identified several genes unique to humans that influence language development. The most famous, FOXP2, shows human-specific modifications that affect neural development in language-related circuits. But it's just one of many genetic changes that helped create our linguistic capabilities. We've identified at least 12 genes with human-specific variants that influence language-related neural development, Dr. Takahashi notes. Together, They help create the neural architecture that makes symbolic communication possible. This symbolic capacity extends beyond language to include all forms of representation, from cave paintings to mathematical notation. Each innovation in representational technology amplified our cognitive capabilities, creating feedback loops that drove further development. Written language allowed us to store more information than any individual could memorize. Dr. Williams explains, mathematical notation let us manipulate concepts too abstract for natural language. Each of these representational technologies extended our cognitive reach. This extension of cognition beyond the individual brain created something unprecedented in evolution: distributed cognitive systems spanning multiple human minds. Modern human cognition happens not just within brains but between them, in the shared symbolic spaces of language, mathematics, and other representational systems. When we solve complex problems today, we're not just using individual intelligence, Dr. Torres notes. We're participating in cognitive ecosystems that include other minds, both present and past, through their preserved symbolic contributions. This distributed cognition allowed humans to adapt to environments and challenges that would have been impossible for any individual intelligence to comprehend fully. From the Arctic to the Kalahari, human cultural adaptations enabled survival in environments for which our African evolved bodies were not specifically adapted. But this symbolic revolution raises a puzzling question. What evolutionary pressures selected for these extraordinary cognitive capabilities in the first place? The traditional narrative of human evolution focuses on environmental challenges, the shift from forests to savannas, climate fluctuations, and changing food sources. But a growing body of evidence suggests that some of our most distinctive cognitive adaptations may have evolved primarily in response to other humans. This hypothesis, known as the social intelligence hypothesis, proposes that the major selective pressure driving primate cognitive evolution was not the external environment but social competition and cooperation within groups. If you think about it, the most complex problems our ancestors faced weren't environmental but social, explains evolutionary psychologist Dr. Eric Torres. Figuring out who to trust, who to form alliances with, who might be planning to harm you. These are computationally demanding problems. Recent mathematical modeling suggests that social dynamics create selection pressures for exactly the kind of general intelligence that characterizes humans. In social groups, success depends not just on solving static environmental problems but on anticipating and influencing the behavior of other intelligent agents. It creates a cognitive arms race, Dr. Torres continues. As group members become smarter, the social problems become harder, selecting for even greater intelligence in a self-reinforcing spiral. This dynamic helps explain why humans evolved general intelligence rather than a collection of specialized adaptations for specific environmental challenges. Social competition requires flexible problem-solving across domains and the ability to generate novel solutions to unprecedented situations. The archaeological record provides some support for this hypothesis. Human brain size increased most rapidly during periods when population density was increasing, creating conditions for more intense social competition and cooperation. Furthermore, the timing of key cognitive adaptations coincides with evidence for increasing social complexity. The emergence of symbolic art and complex technology approximately 50,000 years ago occurred during a period when human social networks were expanding in scale and complexity. We see evidence of long-distance trade and resource sharing emerging around the same time as symbolic expression, Dr. Williams notes. That's consistent with the idea that social challenges were driving cognitive evolution. This social selection hypothesis helps explain another puzzling aspect of human cognition, our elaborate self-consciousness. Humans spend extraordinary amounts of cognitive resources monitoring and evaluating our own behavior and mental states, capacities that seem excessive for solving purely environmental problems. Self-consciousness makes perfect sense in a social context, Dr. Wallace explains. To navigate complex social environments, you need to understand how others perceive you and how your actions might influence their behavior toward you. This social model of cognitive evolution suggests that many of our most distinctive mental capabilities evolved primarily to help us succeed in what we might call the ecological chess game, the complex dynamics of competition and cooperation within human groups. If this hypothesis is correct, it means that human intelligence evolved not primarily to master the external world but to navigate the social world, to influence, understand, and coordinate with other human minds. Our technological mastery of nature may be, in evolutionary terms, a beneficial side effect of adaptations selected for social success. This perspective reframes our understanding of what makes human cognition unique. The critical adaptation wasn't just increased processing power but a specific kind of intelligence specialized for social dynamics, for understanding minds rather than just physical systems. We're not just generally smarter than other species, Dr. Torres emphasizes. We're specifically adapted for thinking about, influencing, and coordinating with other minds. That's our evolutionary superpower. This social intelligence perspective connects with another remarkable human adaptation, one that operates not at the level of individuals but at the level of groups. In 2010, researchers at MIT conducted a series of experiments on what they called collective intelligence, the ability of groups to solve problems together. Their findings challenged conventional wisdom about group performance. Contrary to expectations, the average individual intelligence of group members was not the strongest predictor of collective performance. Instead, the most successful groups showed high levels of what researchers called social sensitivity, the ability of members to accurately perceive and respond to each other's emotional and cognitive states. These findings point to something remarkable about human cognition, explains social psychologist Dr. Veronica Wallace. We're adapted not just for individual thinking but for thinking together in ways that can exceed what any individual could accomplish alone. This capacity for collective cognition depends on a suite of adaptations that facilitate coordination. These include our sophisticated theory of mind, our elaborate communication systems, and our unusual emotional sensitivity to social feedback. Humans are exquisitely tuned to social signals. Dr. Wallace continues, we unconsciously synchronize our movements, speech patterns, even our breathing when interacting with others. This synchronization creates the foundation for coordinated action. Neuroimaging studies have revealed that when humans engage in cooperative tasks, their brain activity begins to synchronize in measurable ways. This neural synchronization facilitates the coordination of attention, effort, and problem-solving strategies. It's as though our brains are designed to form temporary neural networks across individual minds. Dr. Chen explains, we literally begin to think in sync. This capacity for synchronized cognition appears to be significantly more developed in humans than in other primates. While chimpanzees and other great apes show impressive cooperation in some contexts, human collaborative capabilities extend to larger groups, more complex tasks, and more flexible role assignments. The evolutionary advantages of this collective cognition were enormous. Groups that could effectively coordinate their knowledge and efforts could solve problems beyond the reach of even the most intelligent individual. From hunting large prey to constructing complex shelters, collective action expanded the range of survival challenges humans could overcome. But perhaps the most significant impact of collective cognition was on cultural evolution. Ideas could be combined, refined, and extended through collective thinking in ways impossible for isolated individuals. The most powerful human thinking doesn't happen in individual brains, Dr. Torres argues. It happens in the spaces between brains, in the dynamic exchange and refinement of ideas across multiple minds. This perspective helps explain a puzzling aspect of human accomplishment. Individual humans don't seem cognitively capable of inventing complex technologies from scratch. No single person knows how to build a modern computer from raw materials. Yet collectively, humans have created technologies of extraordinary sophistication. We've developed cognitive specialization, Dr. Thompson explains. Different individuals master different domains of knowledge, allowing collective knowledge to far exceed what any individual could comprehend. This collective cognition perspective reframes our understanding of human intelligence. The critical adaptation wasn't just enhanced individual brainpower, but the ability to form collective brains through social connection and symbolic communication. We're the species that thinks together, Dr. Williams summarizes. That's our unique evolutionary innovation. This collective dimension of human cognition connects to another remarkable human adaptation, one that operates not within our bodies but between them. In 1872, Charles Darwin published The Expression of the Emotions in Man and Animals, documenting the similarities in emotional expression across cultures and species. This work laid the foundation for understanding the biological basis of human emotions. But recent research has revealed that human emotional systems contain subtle specializations that distinguish us from even our closest relatives. Human emotions aren't just animal emotions with greater intensity or complexity, explains effective neuroscientist Dr. Maria Gonzalez. They contain specialized adaptations for social coordination and group cohesion. One striking example involves distinctive human emotions like guilt, shame, and pride, emotions that appear to have evolved specifically to regulate social behavior within cooperative groups. These emotions essentially function as internal enforcement mechanisms for social norms. Dr. Gonzalez continues, they motivate adherence to group expectations even in the absence of external monitoring. Neuroimaging studies have identified specialized neural circuits involved in these social emotions. The human anterior cingulate cortex, for example, shows unique patterns of activity during experiences of guilt and social rejection. These patterns differ significantly from those observed in other primates exposed to similar situations. Even more telling are the biochemical mechanisms underlying human social emotions. The neuropeptides that regulate emotional bonding, oxytocin and vasopressin, show subtle but significant differences in their receptor systems compared to other primates. Human oxytocin receptors have structural differences that alter binding properties and downstream signaling, explains Dr. Patel. These molecular modifications change how strongly and under what circumstances social bonding occurs. These specialized emotional systems help solve a critical adaptive challenge: maintaining cooperation in larger groups where direct reciprocity becomes difficult to track. By internalizing social norms through emotions like guilt and pride, humans could maintain cooperative behavior even in large groups where individual interactions might be infrequent. Our emotional systems essentially create bridges between individuals, Dr. Wallace explains. They synchronize behavior and motivation across group members, facilitating collective action. This emotional synchronization becomes particularly evident in shared ritual contexts. Across cultures, humans engage in synchronized singing, dancing, and movement that triggers cascades of neurochemicals promoting group bonding and identity. When people move in synchrony, their brains release endogenous opioids that create feelings of connection, Dr. Gonzalez notes. This biochemical response to synchrony appears to be particularly strong in humans compared to other primates. These emotional adaptations created something unprecedented: the capacity for individuals to identify with abstract social groups extending beyond direct personal relationships. Humans can feel emotional connection to tribes, nations, or ideological communities composed mostly of individuals they will never meet personally. This expanded circle of emotional identification was revolutionary, Dr. Torres explains. It allowed cooperation to scale beyond the limitations of personal familiarity, enabling the formation of societies comprising thousands or millions of individuals. The adaptive value of these specialized emotional systems becomes clear when we consider the survival advantages of large-scale cooperation. Groups that could coordinate effectively could defend territories, share resources during scarcity, and accumulate specialized knowledge across generations. Our emotional adaptations are what allow human societies to function, Dr. Wallace emphasizes. Without our specialized capacity for social emotions, large-scale human cooperation would collapse. Yet despite their critical importance, these emotional adaptations often operate below our conscious awareness. We feel guilt, pride, or group belonging without necessarily recognizing the sophisticated evolutionary engineering that creates these experiences. This pattern of invisible sophistication raises a broader question about human evolution, one that might change how we think about our species' place in nature. The traditional narrative of human evolution emphasizes visible adaptations, our upright posture, our dexterous hands, our large brains. But as we've seen throughout this exploration, many of our most important adaptations operate invisibly, below the threshold of conscious awareness or casual observation. We've been looking for human uniqueness in all the wrong places, Dr. Takahashi suggests. The most profound adaptations aren't visible to the naked eye. They're in our biochemistry, our neural connectivity patterns, and our social coordination mechanisms. This pattern of invisible adaptation raises an intriguing possibility. What if human evolution has been mischaracterized as primarily a story of physical transformation, when it's actually a story of process innovation? Physical structures get all the attention, Dr. Thompson observes. But the real evolutionary magic often happens in processes, in how systems operate rather than just their visible architecture. This process-focused perspective helps explain why humans appear physically similar to other primates despite our dramatically different capabilities. The critical adaptations weren't primarily in our visible anatomy but in the invisible processes that govern our development, cognition, and social coordination. Consider human development as an example. Externally, human development might appear simply as a slowed version of typical primate development. But this external appearance masks sophisticated internal changes in developmental timing and sequencing that create entirely different outcomes. It's like comparing two factories that look similar from the outside, Dr. Williams explains. But if one has dramatically more sophisticated internal processes, it will produce qualitatively different outputs despite the external similarity. This emphasis on process innovation over structural change appears repeatedly in human evolution. Our digestive tract resembles that of other primates in gross anatomy, but the invisible biochemical processes operating within it differ in ways that dramatically expand our dietary options. Our brains share the same basic architecture with other primates, but the developmental processes that construct them and the molecular mechanisms that regulate their function contain human-specific innovations. Most significantly, our social coordination systems, the invisible processes that allow humans to think, feel, and act collectively, represent evolutionary innovations that have no clear parallel in other species. These systems create capabilities that emerge at the group level rather than residing in any individual. We've been looking for human uniqueness inside individual humans, Dr. Torres suggests. But some of our most distinctive adaptations exist between humans, in the invisible processes that connect and coordinate our minds. This perspective on human evolution carries profound implications for how we understand our species' place in nature. Rather than seeing humans as simply improved apes with bigger brains and better tools, it suggests we represent something qualitatively different, a species whose individual capabilities are amplified and transformed through unprecedented social connection. The story of human evolution isn't just about what happened within our bodies, Dr. Williams concludes. It's about what happened between them, the emergence of new forms of connection that created capabilities beyond what any individual could possess alone. This broader perspective brings us back to where we began, with the question of humanity's hidden upgrades. The most profound evolutionary adaptations might be those we take for granted, the invisible processes that make human life possible without announcing their presence. From the molecular mechanisms that regulate our oxygen delivery to the neural systems that enable our social coordination, these hidden adaptations work silently in the background, creating the conditions for human possibility. They are the invisible architects of human potential, the unsung innovations that made our species' extraordinary journey possible. And perhaps that's the most remarkable thing about human evolution. its greatest masterpieces remain largely invisible to those who carry them. We live our lives supported by biochemical and neural innovations of staggering sophistication, yet we rarely pause to marvel at their design. In that sense, the greatest hidden upgrade of human evolution might be our capacity for wonder itself, the ability to step back from our immediate experience and recognize the extraordinary in what we previously took for granted. Perhaps by understanding the hidden sophistication of our evolutionary design, we might develop greater appreciation for the remarkable systems that make human life possible. The human body isn't just an assembly of parts. It's a masterpiece of integrated processes operating across multiple scales, from the molecular to the social. In the subtle engineering of these processes, rather than just in our visible anatomy, lies the true story of what makes us human. Every breath you take, every emotion you feel, every thought you think depends on evolutionary innovations so elegant they disappeared into the background of your experience. They are the canvas on which human life is painted, invisible precisely because they work so well. Our evolutionary inheritance isn't just in our bones and muscles, but in the silent symphonies of molecular interaction that animate them, hidden upgrades that transformed a primate body into something capable of contemplating the stars.